All right, in 6.3, we're going to talk about microbial nutrition. This isn't just what they eat, but how they eat. They don't necessarily really eat. They don't have mouths, but what nutrients do they need? But how do they obtain these nutrients? And energy is another part of this. So we're going to talk specifically about some ways to define different strategies that microbes use. And we'll talk a bit about two cycles of elements, nitrogen and carbon, that globally are very important, that microbes, they maintain these cycles that really ultimately allow us to survive. So what are the limits on microbial growth, right? We said that one E. coli can divide into two every 20 to 30 minutes in optimum conditions. So that means one cell can get to 100 trillion cells in about 24 hours. That would weigh 1 gram if you dried it down. In 48 hours, it would be 10 to the 14th grams. That's approximately 4 million dump trucks full. And ultimately, if there were no limits, microbes would overwhelm us in a matter of days, right? So obviously, that doesn't happen. Microbes have a large number of limits on their growth, and so a lot of that is availability of nutrients. So we talk about what we call environmental niches. These are places to live, but really we also need nutrients in those places. So there are essential nutrients. These are the compounds that microbes cannot make for themselves. They have to get them from the environment. And at a base level, a lot of these are elements: carbon, nitrogen, phosphorus, hydrogen. Don't memorize this, but we also have things like iron, magnesium, and then the trace elements: cobalt, copper, zinc, things like that. These are very similar to what you need, right? Because bacteria and microbes are made up of the same things as you. In the lab, we can provide these things, right? We talked about chemically defined media, only what they need in those minimal mediums. For E. coli, this could be those elements listed, plus like glucose as a carbon source and an energy source. So you'll notice they need carbon, right? Because they're made of carbon. They got to build things out of this carbon, but they also need energy. And glucose can be both of those. It's a carbon-containing molecule, and it has energy stored in it. So here's something that's like a complex media that's not well defined. It's called lysogeny broth or Luria-Bartani broth, LB as it's commonly known. This is just a common thing. It's got what's called bactotryptone, which is some bits of amino acids in there. Bacto yeast extract. So these aren't very well defined, right? They're kind of ground up bacteria / yeast. And this is salt, and it's pH to 7. It's good for growing things. This is a very well-defined medium, M9 medium. It's got these specific chemicals and specific amounts in it. It really depends on what you're trying to grow. And the long and short of it is we don't always know what different microbes need. It's that we know that different pathogens need, because we've studied those very, very well. Some pathogens, some microbes, need extra growth factors that are added to the culture media before they will grow. This can be really useful in identifying specific microbes. So, I don't know, something like Haemophilus influenza needs extra hemin. That's a bit from blood and NAD added. That's why it grew on the chocolate agar, but not the blood agar, because that hemin wasn't released. Legionella pneumophila needs extra cysteine. There's various different things in here that can be added, but these growth factors can be used if you add or remove them from two different plates. You can tell, is this, a specific microbe here? Evolutionarily, how do the microbes survive? they need these things. Where do they get it from? Well, in the case of pathogens, they get it from their host. So they've really lost the ability to make this because it's so readily available in their host. They just steal it from them. Why make it when you could steal it from someone? That's kind of the long and short of it for bacterial metabolism here. In some cases, there are microbes that we don't really know what they need to grow. Complex things like these rickettsia, which actually grow inside of a host cell, we don't know what they need. And we don't have ways to grow them in artificial cultures. So if you want to study rickettsia, you have to grow it inside of cells. And that's tricky, but that's what it's necessary because we don't have a way to artificially grow it in a liquid culture. So one of the key things that has to happen is growing organisms have to obtain carbon. So remember, all organic molecules contain carbon, and they're essential. These carbon compounds, they could be very simple, they could be more complicated. They're used as food, which is going to give energy, but they're also used as building blocks, right? They get broken down and they form the building blocks that you can put together to make other things. We call this biomass. That's the mass, the large amounts of stuff that gets made inside of a biological organism. And there's two different ways that organisms can obtain their carbon. Autotrophs can make their own carbon compounds by just starting with CO2, carbon dioxide, from the atmosphere. They can take that carbon dioxide, stick them together, and start building carbon compounds. So auto means self-troph feeding. So they basically can feed themselves from CO2. Think photosynthetic cyanobacteria or something like that. They can take CO2 from the atmosphere, build it into sugars. The other strategy is heterotrophy. Hetero means other, troph, so you feed off others. Heterotrophs have to obtain their carbon compounds from other organisms. So heterotrophs generally have to eat autotrophs or something that has eaten an autotroph. So this is you. You are a heterotroph. It's also E. coli. They can't do photosynthesis. They have to obtain carbon from other sources. And in the process, heterotrophs often release CO2. So you might see kind of a cycle occurring here. We'll come back to that in a moment. So this is the real simplified picture of it, but it's too simple. There's a few things it's missing. But ultimately, pretty much all the energy on Earth is coming from the sun. That's going down, and autotrophs are taking sunlight and carbon dioxide and sticking carbons together to make sugars. Those sugars get stored there. The autotrophs can use those sugars, and they can keep going. Along the way, some things like you, me, and this goat evolved, and we're kind of parasites off the autotrophs. We have to eat autotrophs to get our organic molecules. Autotrophs don't really need us, okay? But we need them. So that's why things like plants and photosynthetic microbes are so critical to our life. Okay, now I've hinted at a cycle here, but this just kind of looks like it flows from here to here to here and then stops. It's not quite true. There is some cycling of things that happen. So if we look at this, we have heterotrophs down here and autotrophs up here. Okay, so the autotrophs, plants, algae, and there's a lot of bacteria. The majority of our producers can be bacteria in nature. They're going to use CO2 from the atmosphere, carbon dioxide, and energy from sunlight to create sugars. So they're going to take those carbons and stick them together. 6 of those, you get glucose. That, in the process, is going to make oxygen, which you might not think about this, but that oxygen, it wasn't there normally. Before there were large amounts of these photosynthetic microbes, that's basically before life, the Earth actually had a very low oxygen content. And it's only through the actions of these photosynthetic organisms that they essentially change the planet. They change the composition of our atmosphere to make enough oxygen for us heterotrophs to evolve. So they make oxygen and carbon-containing sugars. Okay, those carbon-containing sugars and oxygen, well, heterotrophs eat the carbon-containing compounds and use that oxygen and break it down into energy that powers their life. In the process, they excrete CO2 molecules. We breathe out CO2 and water through that process. That feeds back in, and the autotrophs suck that up and keep going. So this is the global carbon cycle, as we call it. Carbon gets moved around through these compounds back into carbon dioxide. Like I said, though, the autotrophs don't really need us. They could get plenty of carbon dioxide because they also break down their sugars. They also excrete carbon dioxide and water as well. We just are kind of parasites off the autotrophs, basically. So this is the less fun pictorial diagram here, right? Let's start with the autotrophs. There's actually two different ways autotrophs can get energy. I've talked about the sun's light, so they absorb light energy and use that to make a bit of ATP and take carbon dioxide and hydrogen and make glucose, which is a sugar. That's called phototrophy, where they use light as their power. There's a small number of microbes that don't need light at all to get their energy. They take minerals and oxidize them, and this is called lithotrophy. It's very rare. We're just going to touch on the surface here. But they get their energy completely differently. They don't need the sun at all. They can live at the bottom of the ocean where there's no light whatsoever, and they can still make energy through this lithotrophy. So these autotrophs make glucose, which then can get stuck together into polysaccharides. Heterotrophs are going to eat that and break the polysaccharides back down into glucose. That glucose is going to go through some processes that we will talk about, metabolism, in the next chapter. It could be fermented. That could be one way of using the energy there. It could go through this, what's called oxidative respiration. That's a better way of extracting more energy out of it. But ultimately, it's going to get broken down into carbon dioxide and water again, which is going to feed back into the cycle. The phototrophs or the lithotrophs, the autotrophs, are going to use that. So you can also use other things, right? It doesn't have to be polysaccharides, but ultimately these sugars are where most of our energy is coming from. So how you capture that energy, right? That matters. We have phototrophy and what we call chemotrophy or lithotrophy. It's not quite that simple, but so how you get your energy, right? We talked about the carbon moving. Now let's talk about the energy. So we've seen phototrophy. We'll talk about what chemotrophy is. We've seen a little bit of it. You got to have energy, okay? Light or chemical compounds is going to provide that energy. So phototrophs can use light as their energy source. That's by definition, photo means light, troph, feeding. Chemotrophs are going to use the energy stored in chemical compounds as the energy. We talked about the lithotrophs. They use inorganic chemical compounds, things like hydrogen sulfide, things like that. Basically, they eat rocks. They get energy from rocks. You, me, and a lot of other things, microbes included, like E. coli, are what are called organotrophs. We get energy from breaking down organic molecules, sugars, like we just saw. So many of our pathogens use carbohydrates, sugars, as both an energy source, but also as a carbon source. And we'll see where that carbon comes in. This doesn't perfectly line up with the heterotroph autotroph thing. All phototrophs are autotrophs, but not all chemotrophs are autotrophs. Lithotrophs are autotrophs. Organotrophs are heterotrophs. They got to get organic molecules from an autotroph. So it doesn't quite line up perfectly with the other thing. They're just different terms. Don't freak out. Okay, so we could break it down into how organisms feed, I guess. Things like carbon source. Where do they get their carbon to build biomolecules? Autotrophs get that from CO2 in the atmosphere that they fix. This process of taking CO2 molecules and sticking them together into sugars, that's called carbon fixation. So they assemble organic molecules. Heterotrophs get carbon by eating other things, getting organic molecules acquired outside the cell. They break those down into carbon. Then there's the energy side of the equation. Phototrophs get the energy from light. Chemotrophs got to break down molecules to get energy. These could be organic molecules, or in the lithotroph case, inorganic. That's where this electron source comes in. That's like a subset of this chemo bit. Litho, inorganic molecules. Organotrophs, organic molecules. So let's look at an example here. We have a cyanobacteria that uses light to fix CO2 into sugars. They can then break down these sugars for energy and building blocks. So how would we classify them? Use this information. Pause the video. Think about this. Okay, so let's break it down. Well, they use light energy, so they're phototrophic, and they're taking CO2 from the atmosphere and fixing it into long carbon chains, so they're autotrophs. We could abbreviate that photoautotrophic. They are ultimately organotrophic, though, because they're breaking down the sugars to use those as an energy source. So they're a bit of both. They are organotrophs, though. Okay. Another key nutrient that if you've ever done any gardening, you might know of is nitrogen. You might not have enough nitrogen in your soil if your plants are struggling. And it's not just plants that need nitrogen. Everything needs nitrogen because remember, amino acids in proteins, they have a bunch of nitrogen in them. And your nucleic acids, your DNA and your RNA, they have a bunch of nitrogen in them too. So there's a global, what we call nitrogen cycle that converts nitrogen between various forms. And this has to happen because the majority of nitrogen on Earth is N2 gas, which is inorganic nitrogen. Makes up about almost 80% of the Earth's atmosphere, but you can't use it. How sad is that? Okay, it's in a form that you can't use for what's called biosynthesis. So bio meaning biology, right? Biological. Synthesis meaning building. So building biological organisms, you got to have nitrogen, but you can't use the stuff from the atmosphere. But some bacteria come to the rescue and they can do what's called nitrogen fixation, where they take that nitrogen gas from the atmosphere, turn it into a form that we can use called ammonium ions, NH4. So things can start to use that to build these proteins and these nucleic acids and other molecules. Other bacteria take these ammonia ions and turn it into nitrate, a different form. And then there's even still that convert that nitrate back to N2 gas called denitrification. It's really not super critical to know each of the steps. I just really want you to know that there's a lot of nitrogen gas in our atmosphere, and we rely on nitrogen fixers, which are primarily microbes. So you've probably heard of like crop rotation, where farmers might grow one thing one year, squash, and then corn, and then peas or something like that. It can be really good for the soil. It's because some plants have a symbiosis, a living together, with certain bacteria that are nitrogen fixers. So peas and legumes, they make little what are called nodules. They're basically little chambers that can house these nitrogen fixing bacteria. So inside of these chambers are the rhizobium bacteria, and they fix nitrogen from the atmosphere, and they give a little bit of that to the plant. So the plant gives them a home, and the bacteria in the home give the plant some nitrogen that they can use. So they take gas, fix it into ammonium, and then the plants can start to use that. And eventually the plants will break down, their nitrogen will get converted back into nitrogen gas, and the cycle will continue. This is just one small example. These aren't actually the majority of them. The majority of our nitrogen fixers are free-living bacteria that do this in the ocean and in the soil and things like that. Okay, so we've talked about some essential nutrients that kind of fuel the growth of all microorganisms. Microbial genomes have evolved in response to this nutrient availability. Some disease-causing bacteria, they've lost the ability to make things because they just steal it from their hosts, right? We can use that to differentiate between different microbes. We talked about some different ways to describe microbes. We have autotrophs that use CO2 from the atmosphere as their carbon source. They stick those together, fix the carbon together. Microbes can get energy through various ways: phototrophy, lithotrophy, organotrophy. Heterotrophs can consume organic compounds, right, that are made by the autotrophs ultimately, and that gets the global carbon cycle going. So know how heterotrophs and autotrophs interact. Make sure you can define heterotroph and autotroph for me too. And then same thing with nitrogen. Nitrogen gets cycled through by the nitrogen cycle. All right, that's it for 6.3.